Hello, good evening. Hello, hello, good evening. So, without wasting our time, this is going to be our second contact hour when it comes to microbial evolution and ecology. Don't forget that we are still at the beginner's mood, and that is why we just have to make sure that we are all at home when it comes to microbial evolution. And today we are coming on two different areas. We will look at the fundamentals of microbiology and the origin of life. If we are talking about life, our concern in this course is nobody, because we are students of a microbiology, is no other animal except the microorganism. So that is why we are going to talk about fundamentals of microbiology. So just as I said, at some point in time, I'm going to stop. I'm very sure that by the time we'll be getting to slide 10, 15, and so on like that, that is at that point you will have to take your notes, your quiz, and you write it down so that immediately after the presentation, you will just make sure that you partake in the test, and that's going to be part of the assessment for you. So, without wasting our time, without much ado about any other thing, let's just proceed as we go on. Now, in this course or in this presentation, we are going to target the five key areas in this same presentation. Just like I did for you yesterday, we might not be able to finish this because it's supposed to be a 3 hours call presentation, and because I actually want you to make um, meaning and understand this concept very very well, because this is still beginners level in microbiology. So I'm going to like shorten it. So I'm going to present a part here today, then later on we will continue. However, don't forget that every part, whether first part, second part, or third part, is going to contain the quiz in which we are going to partake in them. Okay, so the first thing is that you have to understand in this presentation. I want you to understand the scope, the wide, how wide the scope and the importance of microbial world. Then I want you to also apply basic chemical principles. Your knowledge in chemistry is not because that is what you call biochemistry, that is the chemistry of life. So I will apply basic chemical principles to microbial life. So everything that takes place, the chemistry of life, for example, addition of hydrogen plus oxygen is what is going to give you hydrogen peroxide, and the breaking down of hydrogen peroxide, the breaking down of hydrogen peroxide into hydrogen and oxygen will actually requires a certain enzyme that is called peroxidase. So only microorganisms that contain this peroxidase as an enzyme, they are the only one that can actually degrade into this. So all these are part of the chemistry of life. Then we will be able to we'll get to a point that you have to compare prokaryotic and eukaryotic cell. We'll look at their anatomy, we'll look at the composition of their cell, and we will now look at the function, because you should know that at this point in time, the structure determines the function. So prokaryotic versus eukaryotic cell, we'll look at the anatomy, we'll look at the function. Then the next one is that we also be able to describe certain metabolism like conversion of glucose into pyruvic acid, conversion of a certain enzyme and the breaking down of certain enzyme into very usable form. So we are going to also learn about the metabolism, the growth curve. We will learn about it, the growth pattern, and how can we use this growth pattern in order to be able to control certain microorganisms, especially when it comes to the issue of diseases and epidemiology. So all these are part of what we are going to talk about. Then also the later part of this course, this presentation, will just focus briefly on the basic principles of what is called recombinant DNA technology, or what we can call the genetic engineering. We will just look at, we'll lay some foundation about it. Honestly speaking, it is very good for you to have a very good foundation so that you'll be able to perform well in that kind of courses. So all the courses in microbiology, they are interwoven. So your microbial biochemistry is applicable in microbial ecology and also in microbial evolution. And don't forget that nowadays, when you are talking about anything biology, if you don't know anything about the genetic materials, manipulation of DNA, if you don't know anything about them, if you are telling them that you don't want to deal with anything molecular, that means you are not going to perform well when it comes to the issue of microbiology. So just like, why do microbiology matters? What are the application of microbiology in Nigeria as well as in at global level? Number one is that we need microbiology in Earth. In Earth, don't forget that Earth is wealth. So if we do not have knowledge about microbiology, it might be very difficult for us to be able to perform well when it comes to certain diseases, when it comes to understanding what is called antimicrobial resistance. That is what is called AMR, antimicrobial resistance. It is our knowledge in microbiology that we use to understanding what is called antimicrobial resistance. Then we will also make use of our knowledge in microbiology to be able to understand, to be able to design, to be able to understand certain things about vaccine and some chemical diagnosis. Some of us might not know that what do we call diagnosis. Diagnosis is just when you a kind of chemical interrelationship. That is what's going to give you diagnosis. Okay. Then, as a microbiologist, as studying microbiology, you will also have very good knowledge in about the environment. And we are talking about environment. We mean what the water quality is going to be very, very important for you. If you allow E. coli, Escherichia coli, or some bacteria in your water, you know what is going to happen. So we also need it in waste management. We need it in understanding what are biochemical cycles. So biogeochemical cycles are those ones like nitrogen generation, fertilizer to us. Then it's also very applicable in food and in food industry, like fermentation of yogurt, garri, and palm wine. Then bioproduction. Now, in the Nigeria context, why? How are you useful? How can you make use of microbiology in Nigeria? Number one, safe water, hospital hygiene, as well as food and safety and local biotechnology industries. So what we are saying in essence is, as a microbiologist, you don't just, some of you will just go to the hospital, go to an hospital carelessly, and you have gone to visit somebody who is feeling so unwell, who has been hospitalized for a long time, and because you are not so careful, you just go and contact and expose yourself to microorganisms, and thereby it is going to make you, these are what we call nosocomial infection. These are infections that you acquire from hospital setting, touching hospital equipment, touching, inhaling part of the hospital environment. So that now, what are the key definitions that you actually need to know as a beginner in microbiology, as a beginner in this course? Number one, wherever you come across microbe, This is a microscopic organism. So microbes, they are also referring to microorganisms. A microbe is a microorganism. So that include bacteria, archaea, fungi, protists, and so on. It also contains the group of the viruses. They are still part of a microorganism. So then you also come across a microbiome. Microbiome, you should master the way I am pronouncing them. Microbiome, this is a collection of microorganisms, collection of microbes and their genes in a certain habitat. So for example, in our guts, in our stomach, so if you want to collect all the population of microorganisms in that environment, they are referred to as microbiome. Then you will also come across the word pathogen. Pathogen. Pathogen is actually coming from microorganism that are capable of causing or initiating a disease in a certain host. So these are pathogens. Pathogens are disease causing microorganism. That is another word. Then the next one is that symbiosis. You also come across symbiosis in this course. What do we mean by symbiosis? This is a close, long-term biological interaction or biological interrelationship between different organisms. Now, I want you to understand something. Syn, S-Y-N, syn, that is similar. Bio, from life, that is bio, symbiosis. So this is a kind of interrelationship or relationship that involve two or more similar, in which the interrelationship or the relationship is benefiting both of them. So that is a symbiosis. So commensals of what is called commensalism. This is talking about the relationship in which microorganism lives with a host with no obvious harm or benefit to the host. Commensals, they live in a host, but they don't have anything benefiting. They do not derive anything, any benefit from that kind of a relationship. So then another one is opportunistic. There are some microorganisms that in their own life they are opportunists. They are usually harmless. Under normal circumstances, they will exist as your friends. They will exist in close proximity with you. They will exist to, they will appear to be your friend, and they will be so harmless. They will mean no harm. They are not they are not in any way affecting you, but there could be certain condition, especially when the immune system of the host is now reduced, is now being tampered with, so they will take advantage. So this from the word opportunist or opportunistic. So you should know that they are not our enemy, but they take advantage. For example, now HIV will reduce your the immune level of the host, and there will be certain microorganisms that naturally they are not your enemy. They will just seize that opportunity and they will start causing diseases when they know that you do not have the ability to fight them any longer. So now we cannot just do this course without talking about history. As a scientist, we are also students of history. You should know where we are coming from. You should know where we are today, and that is going to give you the idea of what and where we are going to be tomorrow. The story of microorganism or microbiology actually started with Robert Hooke and Anton Leeuwenhoek. They are early microscope, that is the first microscope, observed and called microorganism animalcules. Animalcules. Animalcules, as at that time, they did not have the right word in which we are referring to them as microorganism today. But what they discovered was, that time, Anton Leeuwenhoek is the father of microscope. He was the first that discovered that you can actually use magnifying lenses, combine lenses together to form microscope. So by the time you now see, they now discover that there are unseen, there are some microorganisms that are too small to be seen with naked eye. But instead of them to regard them or to refer them to as a microorganism, they call them animalcules. Animalcule, that is particular particles of animal. So animalcules. Then this will also continued unto the time of Louis Pasteur, who disproved this theory of spontaneous generation. So at that time, the scientists believed that life can actually come from non-living. That is, you can have living animals or living particles coming out from none. No? So it was Louis Pasteur that actually disproved, discontinued, brought up certain experiments to neutralize or to kick again, to kick out the theory of spontaneous generation. Louis Pasteur also introduced us to the process of fermentation up to today, that we are still using today. So it was Louis Pasteur. And he also advanced knowledge in vaccine biology. So it was during the Pasteur that we actually came about what can be regarded as a vaccine. Then Robert Koch, he was the one that talked about pure culture. Koch postulate. He was the one that first of all linked or associated certain diseases to be as a result to be caused by microorganisms, to be caused by organisms that are too small to be seen with naked eye. And that is the reason why up to today we are still making use of this opportunity and this idea, and that is a pure culture. Now the next thing is that in this course you should know, and we are going to see it, that you can actually group or classify microorganisms into three distinct kingdom or three distinct pathway. Okay, the first one, which they call bacteria and archaea. The group of bacteria and archaea contain prokaryotic cell without membrane bounded organelles. So all these one will form what is called prokaryotic cell. We are going to see it. Then can also be divided into eukaryotic microbes, which include fungi, yeast, or mold. Then protists. Protists contain is made up of the group of algae as well as protozoa. Then there are some acellular agents, acellular, that is absent. We cannot actually regard them as living, and that's why virus up to today, scientists still believe that virus can neither be created and we can neither be classified as living as well as non-living, because outside the living cell they will exist as a particle, as a non-living, but once they are inside the cell, that is when you will see them causing havoc. So you can actually group them into that group that is called acellular, that is acellular, which consists of the group of viruses, virions, and prions. So we will see and we will know more about this as we go on. Now, I told you that as a biologist, you need to also understand the chemical principles that involve atoms and bonds, how bonds are being formed, covalent bonding, ionic bonding, hydrogen bonding, van der Waals forces of attraction, because you can never understand what is even called the materials, the DNA and some molecules of life, they are coming together to form some chemicals, compound in form of by coming together with certain bond. So biological atoms like carbon, hydrogen, oxygen, nitrogen, phosphorus, as well as solvent, they dominate. So everything, if you collect everything in this life, just collect them. Think of either aquatic, think of anywhere, you just discover that you can synchronize them into containing carbon, hydrogen, oxygen, and all these biological atoms. For example, when you pick up carbohydrates, when you pick up protein, carbohydrate is made up of carbon, hydrogen, and oxygen only. And when you are talking of protein, that is when nitrogen will come in. That is when phosphorus will come in. That is when sulfur will also come in. So these biological atoms, they are the basis, they are the foundation of life. They are the foundation at which, so wherever, if you talk of microorganism, if you talk of any, so these are the atoms. Then water, we also talk about, you see, let me just say that at this point in time, you will know that life cannot occur, but life cannot take place without water. And that's why in evolution, we still believe that even human beings, we must have migrated, we must have descended from, because 90% of our blood is water. So we have to understand that water is very special, in terms of their polarity, in terms of hydrogen bonding, and in terms of their property as a universal solvent. And we have to also appreciate them when it comes to the power of cohesion. Okay, now general instrumentation. We made mention about pH. So pH is a measure of alkalinity or acidity. So, and a certain microorganism, that is, they have optimal pH in which they can exist. So, for example, acidophilic, that is telling you that these are microorganisms that can survive in acidic condition. And when you are talking about whenever the pH is tending towards neutrality and is tending towards alkalinity, you now discover that they won't be able to thrive well. So when you pick up a microorganism, for example, it is going to have its own optimal pH ranges in which it can exist. And anything outside that, that is when you see their growth is being affected, and that's when you will now see that the metabolism is affected, their growth is affected. Then we will also talk about buffer resist pH change. What do we mean by buffer? Buffer is something that balances. I told you that in general instrumentation class yesterday, that what do we mean by buffer? Standardize. We need to standardize. For example, we have to compare our sample. We have to compare it with a standard, with a reference. So if you are picking up a microorganism and you are getting the pH of that microorganism, you have to compare it with a standard. So that's why the pH, acidity or alkalinity, we'll be talking about whether or not it is going to be. So we compare it 4, 7, as well as 10. So you need to know that. Then macromolecules like carbohydrates, lipids, protein, nucleic acid. These are macromolecules, that is, molecules that exist together, molecules that join together in order to make sure that life continues. Now, enzymes. What are enzymes? Enzymes are biological catalysts that speed up the rate of biochemical reactions without taking part. So, time will fail us. I want you to go back to your elementary chemistry, whereby you go and read more about biological catalysts or catalysts. They alter the rate of chemical reaction and they themselves, they are not taking part in that reaction. Then again, enzyme, they have their own active sites, and they are very, very specific. Then you also come across what are cofactors and coenzyme, co-colleagues, cofactors, coenzyme, enzyme, the colleagues of enzyme. They are not enzyme directly, but they can collaborate, they can co-exist, they can take part in certain biological reaction. So then temperature and pH, which affect enzyme activity. So that's very, very important. You know, enzymes are proteinous in nature, and anything that is going to bring about increase in temperature, that means that enzyme, the protein, will be denatured. So you have to take note of that. Okay. Now, this is a typical prokaryotic cell. So we can lecturer design of a prokaryotic cell. Prokaryotic, it can be divided into three based on the composition of their cell wall. When the cell wall is like this, look at this. You can see that this one are called gram positive. And why are they called gram positive? Because they have a thick peptidoglycan. Thick, you can see. This is the bluish parts are called the thick. They are called peptidoglycan. And the gram negative, the blue, are very, very thin. Very, very thin. And that's why they call them negative. Gram negative. Negative, thin. Positive, thick. So if somebody, even if you don't know any other thing, differences between gram positive and gram negative. Negative is having very thin peptidoglycan plus outer. So just remember that very thin peptidoglycan. The thick one is a positive, that they have very thick peptidoglycan. And there are some bacteria that they or prokaryotic cell that can neither be classified as either gram positive or gram negative, but they are classified among what they call mycobacteria. An example is a Mycobacterium tuberculosis. And why? Because instead of them, in addition to peptidoglycan, they now have another thick. So what you should just, I want you to just know about the cell wall, about the prokaryotic cell structure, is that number one, thick. Somebody who is putting on very thick coats and somebody who is putting on very thin, you will know and you will agree with me that the one that has a very thin will be easy to control, will be easy to catch cold, rather than the one that contains, this one is thick already. And in addition to even being thick, some microorganisms, they also have the presence of waxyl mycolic acid. So that is, in addition to peptidoglycan, they also, so it's just like when you have somebody that is putting on very thick coats. Apart from that, the person is also wearing cardigan, is also wearing a lot of things, I mean, lot of other clothes, in order to make sure that it is not catching cold. So and that's the reason why members of Mycobacterium group, they are very, very difficult to control. That is why you see them, they have that high ability to be able to develop resistance against all these common antibiotics. So that is one thing you should know about the architectural plan of the cell structure. So the first thing is the cell envelope. You know what they call envelope? So the cell structure can be divided into three. Let me just mention that. So the first one is the cell envelope. The cell envelope is what contains the cell wall, and that is what classifies them into either gram positive, gram negative, or we also classify them into what? Into whether they are Mycobacterium tuberculosis or not. And that's why gram stain. That is why gram stain cannot be used in order to diagnose Mycobacterium. So they have to. So the first thing is that you have to use, that's why you have hydrochloric acid as part of the stain. The first thing is that they have to look for an acid that's going to disrupt and destruct the mycolic layer before it will now be stained with common stains. Okay, so after that, the prokaryotic cell structure is made up of cell envelope, made up of the cell internal features. And what are these internal features? Internal features include ribosomes. Ribosomes, 70S and 30S. You will get to know all this when you go higher. Then endospore, that is, which is very, very common in Bacillus and Clostridium tetani. So this is very, very common. There are some microorganisms that apart from having the cell envelope, which is the external, so they also have very thick endospore. Endospore. Then the next one is a cell surface structure, that is, on the surface here there are some bacteria or prokaryotic organisms that they also have that. And you can see like this one is having flagella, and there are some that will have biofilm. All this will help them to be able to attach themselves to the surface in which they exist. Glycocalyx. We are going to see most of these things by the time we go on, by the time we go higher. Don't forget that this is just a beginner class in microbiology. Now, another thing, just like the addition to what I have said so far, is that gram-positive contain thick peptidoglycan. And you can see it. It is more prominent here. This blue is what is called peptidoglycan. And this one, look at this one now. You can see that this one, this red one, you can see the gram negative. If you look at this one, this is the color of this one, that is membrane. This one now, that is peptidoglycan. So gram negative has very thin lipopolysaccharide, very thin. It has it as the outer membrane and the inner membrane. So, but the third one, which are called the acid-fast group, this one contains mycolic acid. In addition to peptidoglycan and some others, they have what is called acid waxy mycolic acid. So that's why they call them what? Call them mycobacteria. Okay. Now, the cell envelope is just another way of understanding what we have been saying so far. You can see acid fast, they have waxyl. You can see waxyl mycolic acid. Apart from having peptidoglycan up and middle, they have waxyl mycolic acid. Okay, so now, then, like I said, the internal features consist of a cytoplasmic membrane, which they use for transport and energy generation. Apart from that, they also have a nucleoid, what is called nucleoids, which are circular chromosomes. Instead of by now, you know the structure of a chromosome in man. Their own is not like that. That of chromosome in man is like X, but their own shape is circular. They exist in plasmid form, circular form. So, and you will be able to see that. Once again, you can still see them still the same way. This is just another structure, another image to explain what we have been saying so far. The envelope is what classifies them into either gram-positive or gram-negative, or whether they are acid-fast. The internal structure, which consists of endospore as well as a ribosome, and the surface appendages, is it going to be in a form of you can see it here. Okay, then prokaryotic cell, just like we have said, you can see glycocalyx, fimbriae, or what is called pili, which they use for attachment as well as a conjugation. Okay, now let's go to eukaryotic cell. What are the differences between eukaryotic and prokaryotic cell? So you can see that I made mention that under prokaryotic cell, pro means primitive. Karyo, that is a nucleus, that is, they don't have nucleus. They don't have anything called organelles. So that is a prokaryotic. But in hue means true. If something is true, karyotic, that is, they have true nucleus and they have what are called membrane bound organelles. All these membrane bound organelles, example include Golgi apparatus, endoplasmic reticulum, mitochondria and chloroplast, and so on and so forth like that. So in eukaryotic cell, they have membrane bounded organelles like nucleus, endoplasmic reticulum, and Golgi apparatus. Another thing that is very, very common in them is that in animal, in some you have mitochondria. In plant life, they are called what? Chloroplast. So, and this is as a result of endosymbiosis. We are going to learn more about what is called endosymbiosis. But at this level, you just know that endo means internal. So they believe that both evolutionarily, they believe that both mitochondria and chloroplast, they are from the same parent. They are from the same ancestor. So they now believe that the bigger one just swallow the smaller one. That is what they call endosymbiosis. And another thing, another very, very prominent structure that you can see in them are cytoskeleton, like HS, ribosomes, as well as cilia, as well as flagella. So you can see the difference between the anatomy of eukaryotic cell as well as that of prokaryotic cell. When you look at the structure here, when you look at this, the typical structure, the architectural structure of eukaryotic cell, you see eukaryotic. So what you see here is nucleus. Is nucleus. And all this one that you can see, the Golgi apparatus, mitochondria, cytoskeleton, all this one, they are called organelles because they are membrane bounded. You can see them. They are covered by a membrane. They are being protected. Their internal are being protected. Their internal structures are being protected by membrane. That's why they call them organelle. Organ, that is small organs. So organelle. So you can see it here. You can even see what they have here, like a ribosome. You can see it that it is coming in form of ribosome. You can see it like this. So instead of them to have 70 years, eukaryotic is having 80 years ribosomes. 80 years ribosome. So that is one of the differences between eukaryotic and prokaryotic. I've told you that in prokaryotic organism, we don't have membrane bounded organelle, whereas in eukaryotic, they have membrane bounded. Apart from that, the ribosome in prokaryotic organism is 70 years. We will know more about 70 years and 80 years by the time we start to talk about protein synthesis. But at this point in time, I want you to know that instead of 70 years, they have 80 years in eukaryotic. So we can see that as we go on. So just like I said, the cell wall and membrane of eukaryotic cell, in fungi, it is made up of a chitin cell wall, like what we have in prokaryotic organism that we are talking about, what classified them into gram positive or gram negative, which is based on the cell wall. Here, their whole is not made of peptidoglycan, it is made up of chitin. It is made up of chitin in fungi. The same cell wall in algae is made up of cellulose or silica. Cellulose or silica. Then in protozoa is an eukaryotic organism. It has no rigid. This one has a rigid cell wall which is made up of chitin. Then this other one in algae has cellulose which is made up of silica. But in protozoa, they have no rigid cell wall. So then they now have pellicle. Instead, no rigid cell wall. So the cell wall is now being replaced by pellicle. So I want you to take note of that. Then in the cell wall and membrane of eukaryotic cell, they are talking of sterols. Sterols. For example, cholesterol in eukaryotic membrane. So you know we don't have all this. Now in a prokaryotic organism like the acid fast, instead of them to have waxy waxy coat, which is called okay, so this one has a membrane that is made up of cholesterol. It's made up of cholesterol. Okay, now you can see eukaryotic membrane, just like I said, this fungi. This is chitin that is made up of the cell was made up of chitin in fungi. That has been replaced with cellulose or silica in algae. Then that of protozoa has been replaced with pellicle. And just like I said, in some steroids, we have cholesterol. Okay, so and what we have been saying so far is that in terms of prokaryotic and eukaryotic, and what do they use each of these for? So you can see that if you are considering the features of a prokaryotic organism in terms of their genome, you can see it is circular, no nucleus. In eukaryotic, it is they have nucleus, but their genome is linear. Then this one is very useful in gene regulation and replication. Then ribosomes, I told you 70 years in prokaryotic, 80 years in the eukaryotic. So, and the difference, this difference make them to be targets for certain antibiotics. Okay. Now, the antibiotic, if you take certain antibiotic like chloramphenicol, it is going to target the 70 years. It's not going to target you because you are an eukaryotic cell organism. So that's the reason why certain drugs will target your own cell, I mean their own cell, not your own cell. Any antibiotic that targets your own cell means that it's going to be poisonous to you. So that is the distinction of into 70 years and 80 years is what now make them to be able to be targeted by antibiotics. Okay, then I told you that organelles, small organs that you still have like mitochondria, chloroplast, endoplasmic reticulum, Golgi apparatus, all these are completely absent in prokaryotic, but in eukaryotic they are very much present. And because they have compartments, they have wall, they have membrane. So that membrane is what is responsible for compartmentalization. Compartmentalization means separation, delimitation, distinction. It is what differentiates the internal from external because they separate them from that. And the cell wall, I told you that the cell wall is made up of a peptidoglycan, in most peptidoglycan, whereas in eukaryotic it is made up of a chitin, cellulose, or it can be present, I mean it can be absent completely. And that's what they use for staining technique to differentiate them into gram-positive, gram-negative, or acid-fast. And they are also target for certain drugs. Okay, then in terms of sizes, you discover that the size of prokaryotic are smaller, is between 1 to 5 micrometer, whereas in that of eukaryotic it is 10 to 100, and it is very responsible for microscopy and diffusion limits. Okay, now I have made mention about this, so I want you to still revisit it in order to be able to compare and contrast the components of prokaryotic cell. So now is the metabolism. So you don't forget that I told you that in this presentation we are still going to talk about metabolism. So anytime you come across metabolism is the big picture of what's happened. the big picture of the whole processes that takes place. And that processes, that's why metabolism is divided into two in form of catabolism as well as anabolism. Catabolism is breaking down. For example, now glycolysis is an example of catabolic reaction. That is why glucose will be broken down into pyruvic acid. Then later on, you talk about cellular respiration, and that's going to be what you have. And anabolism, we'll be talking about building up, building up. And ATP will be regarded as the energy currency. Nigeria has its own currency. Every country has its own currency. Dollar, American dollar. So the currency of the whole cell, the acceptable currency, the best form of currency that you can talk about, that will produce, that will help you as a medium of exchange between catabolic reaction and metabolic reaction, is called the ATP. That is adenosine triphosphate. So that. Then we will also look at redox carrier in form of NAD/NADH, FAD, and so on. All these are part of what we call coenzyme. We are going to see more of this as we go on. Okay. So without wasting our time, I know very well that our time is fast spent, and that's the reason why that I won't be able to go further from here. So I want you to take this quiz. Take this quiz. That's the first quiz. So the first quiz is multiple choice question. That is the objective. Which statement about enzyme is most accurate? So which is most accurate? We have to pick either A, B, C, or D. So, and the next one is you have to explain how temperature affects the pH. How temperature and pH affects the performance of enzymes or the function of enzymes. So you might not see it, but explain how temperature and pH affects the performance of enzyme. Okay. Now, apart from that, you have to also do the second question is cell wall. Yes, take this as second question. Which pairing is correct for eukaryotic microbes, fungi, cellulose? So then you have to compare the functional roles of steroids in eukaryotic membrane with organoids in some bacteria. Then why do these differences matter for drug targeting? So I want you to take note of that. Take note of that. Okay, that's a second question for today. The third question now, I want to move to the third question. Don't forget that you are to do it immediately before the class closes. So you have to do it before the class closes. Once I log out, that means I may not be able to take it any longer, and you have to submit. You don't need to write your number anywhere. Just do it on your comment section. Submit it in comment section on YouTube. I'm not asking you to submit in WhatsApp. I'm asking you to submit in comment section. And don't forget that the next time we'll come back about this course, we'll be talking about metabolism. We'll start again from metabolism, which is which I call the big picture, the big picture of everything. So the third question is fermentation versus anaerobic respiration. So fermentation differs from anaerobic respiration because to pick which one is correct. And any option you are picking, you must don't just write A or B. No, no. If you are picking A, you write A, you write uses oxygen as the terminal electron acceptor. You put it there. Don't put not only the option. So the next essay question is using lactic acid fermentation as an example. I want you to outline how NADH plus is regenerated into and discuss two industrial or clinical application Okay, so don't forget this is the third one. The first one is this enzyme, the basics. Okay, this is the second question, and this is the third. That is the third. So please, I am giving you only 25 minutes to do it. Only 25 minutes to do it. Okay, 25 minutes to do it. Prokaryotic cells are going to see are also be divided into eukaryotic microbes, which include fungi, yeast, or mold. What is contained Please continue working. This is the third slide on your quiz. The third slides on your quiz. This is the second one. That is the second one. That is the second question. And this is the third question. This is number one, number two, number three. One, we have to explain how temperature and pH affects the performance or the activity of an enzyme. We have about 15 minutes more, about 15 more minutes. Hello, ladies and gentlemen. We are coming to the end of today's lecture on microbial ecology and evolution. Don't forget what we have said so far. Don't forget that you are to submit immediately as I am logging out. Let me know in the comment section. Let me know your response. Let me see your response in the comment section. So, in order not to just leave you, in order not just to abandon you here because of one thing or another, so that you'll be able to do it very, very well. I mean, the quiz. I want you to let me end my live streaming. Let me end it here, and I want to believe that you should keep your responses, keep it coming in. Yeah, I want to be so flexible. Don't let me be too rigid. So let me allow flexibility to come in, and the flexibility will be in time. So with respect to the time, so please do it and submit. Do it and submit. Do it and submit. So it is on that note that I want to thank every one of you for being my special guest in today's presentation. It has been so wonderful. Apart from the question, if you have any question on any query, let me know in the comment section. Let me know in the comment section. You can put any question across to me in the comment section. I'm going to respond to it.